The circadian clock in mammals keeps time with the 24-hour rhythm in nearly every cell in the body. This molecular clock drives the expression of genes to peak at specific times of the day. In part 1 of this video tutorial, we will explain how the circadian clock controls gene expression and give an example of a gene that is clock controlled and affected by circadian disruption. In part 2, we will explain the complex regulation of this gene in more detail and show how the clock and metabolism are intertwined. The core of the mammalian circadian clock is a feedback loop composed of proteins called transcription factors that bind specific segments of DNA called response elements. Once bound, transcription factors can turn on or off the transcription of the downstream gene. In the mammalian circadian clock, the transcription factor called clock for circadian locomotor output cycles kaput dimerizes with another transcription factor called BMAL1 for brain and muscle aren't like one. The clock BMAL1 dimer binds to the subtype of response elements called enhancer boxes, or E-boxes, and promotes the transcription of cryptochrome 1 and 2, abbreviated CRY, and period 1, 2, and 3, abbreviated PER. After transcripts of PER and CRY are translated in the cytoplasm, the proteins dimerize, return to the nucleus, and interact with the clock BMAL1 dimer. This interaction interferes with the ability of clock and BMAL1 to stimulate transcriptions of PER and CRY genes. Thus, new transcripts that encode PER and CRY stop being made. As PER and CRY proteins degrade, clock and BMAL1 are free to promote PER and CRY transcription again. The cycle starts again, forming a negative feedback loop that generates 24-hour cycles of gene expression that affect behavior and physiology. The clock BMAL1 dimer also promotes the expression of other transcription factors called ROR alpha, beta, and gamma, and reverb alpha and beta. These transcription factors control BMAL1 transcription at ROR response elements called ROREs. The reverb protein represses BMAL1 transcription, indirectly controlling its own transcription. Reverbs can also bind to a response element called the destruction box, or D-box, and control expression of other genes. Meanwhile, RORs promote the transcription of BMAL1 and cause BMAL1 proteins level to rise. Reverb and ROR act as an auxiliary loop to reinforce the core circadian clock and to help maintain its robust oscillation. These transcription factors and response elements affect the circadian clock and the expression of many additional target genes whose promoter regions have E-boxes, ROREs, or D-boxes. These other genes that have clock-dependent expression are called clock-controlled genes. One clock-controlled gene is NAMPT, which encodes nicotinamide phosphoribosyl transferase, an enzyme that is critical for the production of NAD+, and therefore for cellular metabolism. NAMPT expression is dependent on the circadian clock. The clock BMAL1 dimer binds to E-boxes in the upstream regulatory region of the NAMPT gene, increasing NAMPT expression. This leads to circadian NAMPT expression. If one or two of the E-boxes in the NAMPT promoter are removed experimentally, clock BMAL1 still promotes NAMPT expression. But when all three E-boxes are removed, NAMPT expression is very low. Thus, NAMPT expression depends in part on the circadian clock. NAMPT also has a D-box. E4BP4 is a transcription factor that binds to a D-box to repress the NAMPT gene. The clock component reverb inhibits E4BP4, which causes NAMPT levels to increase. When the circadian clock is disrupted, NAMPT expression decreases and becomes arrhythmic. This has been shown in many different tissues and many different circadian clock mutant models. For example, in isolated fibroblasts grown from CRY1, CRY2 double knockout mice, scientists found that NAMPT expression is low and stops oscillating. This experiment also shows that the clock regulation of NAMPT occurs within the cell and is not dependent on the other clocks in the body, such as the suprachiasmatic nucleus. In mice without reverb, the E4BP4 repressor of NAMPT is high, which results in low NAMPT expression in the livers of reverb knockout mice. Similar results have been found for clock delta 19 and BMAL1 knockout mice, which no longer have the clock BMAL1 dimer to drive NAMPT expression. These studies show that NAMPT expression is dependent on the circadian clock function in many different ways and within the same cell. It is estimated that in a given tissue, 10 to 50% of genes are clock-controlled in mammals. NAMS is an example of a gene controlled by the circadian clock that is critical for cellular metabolism. This is just one example of the ways that the circadian clock can control gene expression that is important for survival.